Welcome back to the MOOC NPTEL course on Bioengineering, an interface with biology and medicine. In the last lecture, we talked about some basic concepts of development. In the same theme, today we are going to continue studying about cell reprogramming, how cell can be reprogrammed, and we have tried to do some many elegant experiments in this area where people have tried to understand how cloning can be performed at animal level and for different type of purposes, both for reproduction as well as from the therapeutic point of view. In today's discussions, in today's lecture, I will continue and talk to you about some successful experiments in the areas of induced pluripotent stem cell making, as well as there are many cloning attempts which has resulted into some of the major breakthroughs in the field. Some of them have resulted into some big scientific groundbreaking research and got Nobel Prize. Some of them have also resulted into the scientific scandals and got some punishment. So, let us continue discussing about cell reprogramming. Alright, let us shift gear and move on to second topic, which is cell reprogramming and cloning. Cloning, I think briefly we talked in the context of DNA cloning, how to make multiple copies of DNA, not in great detail, but I was talking to you about role of biotechnology, in which way you can make multiple copies of the genes, which could be favoring certain properties that can be used for the recombinant DNA technology. I had talked to you briefly about it. Let us look at some of the animal and organism cloning part, and this part looks much more kind of interesting, fancy, scientific fiction type of idea. But before that, even cloning also can happen in the plants, because plant cells have much more ability to make totipotent cell. It means the cells have the property which they can de-differentiate to any type of the cell if you just provide them right hormone and right medium conditions. Let us see this experiment. This is a carrot. If you are taking the small piece of the carrot root, putting in some nutrient medium, you can see that the cells are dividing, and now a small embryo is developing from it. And if you put now it in again some nutrient medium which contains cytokinin and gibberellic acid, etc., those hormones, now it can form the root and shoot, and it can result into the full carrot. So, plant cloning, of course, is very useful from agriculture point of view. Many times you would have seen that people have taken a small part of a given plant and may use those plants to now further grow and put in a different field, right? And they actually result into new trees or new plants. So, there are in fact some plants which I do not know whether you have noticed any or not, like if the leaves even fall down from them, and if from the surrounding of those soil area, even those leaves will result into certain type of another tree. An example? Right. So, some of the banyan tree and many of the other examples are there which do follow this kind of pattern. And cloning in general, plant cells are much more flexible, having much more totipotency in that way that they can modify themselves, and they could actually result into the full new plantlets. So, if you have not cloned a plant, of course, that is something, a small experiment to be tried out, which you can do yourself. It is not going to harm you. Let us think about the animal cloning, which is much more tedious process. Many things has to be controlled for a full animal to be made if you want to try it out. So, nuclear transplantation is one of the concept which aims to look at whether similar type of plants, type of concept can be used for the animal cell. Are the animal cell totipotent? Can they be converted into any type of cell which you want? So, scientists have tried to do the experiments. They use some of the unfertilized eggs as well as some of the fertilized eggs that they took. They try to replace it with the nucleus of the differentiated cells, which are much grown cells, and then try to do the experiment to test out the hypothesis, can the nuclear transplantation may happen. So, the possibilities are from this hypothesis that if nucleus from the differentiated donor cell retains its full of the genetic information, then it should be able to direct the development and give rise to any type of tissue type. It might be very complex right now. Let us think about this hypothesis in much more schematic form. Think about experiment which was done in 1960s. Intention was to look at whether animal cells have the similar type of totipotency properties, and what type of animal cell may have those properties. So John Gurdon, he did experiment in 1960s using frog eggs. So he took the frog egg cell and did the UV radiation to remove the nucleus from this frog egg. Now what you get is enucleated or nucleus removed from the egg cell. So just pay attention to this experiment and then you will be clear about the experiments. So now you have this enucleated egg cell and you are trying to add a nucleus, you are doing the nuclear transplantation from one of the two conditions. Condition one is you are taking an early stage of the frog embryo, taking some cells from it, and now you are trying to use this particular egg cell environment and use those nucleus to fuse the thing together and see can it now develop into the full embryo. That's one possibility. Second possibility, they took the developed cells, like a skin cell or intestinal cell, not the embryonic cell, but the developed tadpole developed cells at the intestine cell layer or the epidermis kind of layers, and from those they took out the nucleus now again and trying to transplant into this particular egg cell again. With this possibility, when they tried out, they only 1 or 2% of them resulted into the successful embryo, and it did not result into the full tadpole. Whereas with this experiment, when they used the early embryonic stage of the frog cells, then it resulted into the successful transplantation, and many of them developed into the tadpole larva. So it just conveyed them it is possible to use the right stage of the animal cell for the nuclear transplantation to happen, and if you can use some of the early embryonic stage cell which are less differentiated cell, then probably they can achieve the totipotency potential. So this experiment was just imagine thought and done in 1960, and with a very limited technology available, with just some good hypotheses and good concepts which they tried out. Based on those, many people have started trying out many type of animal cloning, right? And let me take help of some of you to read rather than I talk about it. Okay, so this cloning of frog was one of the very successful experiment being done, and many people started following, can we do animal cloning on different type of animals as well, because frog is not something which can give you anything commercial output. Think about, can you make cloning of the dogs, cloning of the cow, cloning of sheep. All of them have a lot of commercial values, right? The high breed of these animals, if you can make multiple copies of them with the cloning process, then probably you can have a lot of commercial values. So many people started venturing into the cloning of animals, different type of animals. And then I am not going to ask you questions about the scientist's name who did that, but just trying to show you the progression that from 1960 onwards, many people have tried to do cloning of animals, and some of those are listed here, like after John B. Gurdon did the cloning of frog. 1986, Dr. Steen Willadsen, they tried cloning of the immature sheep. 1994, they tried the cloned calves, Dr. Neil First. And 1996, Dr. Ian Wilmot of Roslin Institute in UK, he did a very successful experiment. First time from the body cell, the somatic cell, they made the sheep dolly. I'm sure many of you would have heard at least, in one of the interesting story format about the success of dolly, right? So this is one of the success example, because you are now trying to reprogram even some of the somatic body cell and trying to develop into the full animal from that. So reproductive cloning of mammals is something which has lot of both commercial aspects, values, and limitations and ethics, and of course danger of going into wrong hands and doing something wrong, right? So it is much of the issue of a debate, but it is a fact. It is people have now mastered the technologies and they can do these cloning processes. So scientists have shown that they could clone mammals by transplanting nuclei from the early embryonic stage. We have talked about the original experiment of Gordon, and then many people have tried in different other animals. From the early embryonic stage, you can take those cells, you can transplant them, and you can use them as for the cloning purpose. However, it was challenging that whether the nucleus from the differentiated cell or the matured cells or the body cells, whether that could be used for the reprogramming. Because even Golden, when he showed the developed tadpole larva from its intestinal epidermis, they were not able to succeed into the cloning process. So this remained still challenging, that whether you can use the normal body cell, the somatic cells, and use those for the nuclear transplantation experiment. So that's where the scientific fiction actually transformed into reality. And the scientist Dr. Ian Wilmot of Roslin Institute, they first time claimed that they can now make the full large animal, sheep, by using the somatic cells. And this experiment has a lot of value because they are first time able to achieve de-differentiation of these donor cells just by taking the memory cell of the sheep. And in many ways, they have mastered understanding about the cell cycle. They know that at which stage you can keep the G1 stage controlled, where you have to make it, before G1 there is a GO stage, when you can, keep the cells in that stage in the dormancy, when you can trigger the factors for it to, now turn into the proper division. All of those were so well understood that now they can play with the conditions, play with the nutrients, and then you can able to culture the memory cell and trying to use those for the cloning. Here Dr. Ian Wilmot is shown, and for your delight, I had also some interaction with Dr. Wilmot when I was at Harvard. So the experiment, what it happened, in this experiment they took one of the sheep from which they took the memory cells. Just took the memory cell, which is, you're not taking any genetic contents of it, you are just taking the, not the gametes, just taking the normal somatic cell, memory cell in this case. And then you took another donor sheep from which you are taking out the egg cell. So from this particular sheep, now you have removed the nucleus out, you got the enucleated cell, and now from the memory cell you are taking out the nucleus. You want to transplant. And for this process, people have tried many things. Sometimes even just an electric shock, can help to facilitate the fusion of this particular cell with the nucleus. And you need many attempts. People, even they have tried probably hundreds of attempts. Of those, only 1 or 2 were successful, which resulted into this kind of cloning. So it's not, so guaranteed process that it always happens. But after many attempts, they were able to succeed into some of the cell which could incorporate and grow into the early embryonic stage. Then they used third surrogate mother, not these two, but a third surrogate mother now, where they have transplanted this embryo, which resulted into the sheep dolly. So it looks like a scientific fiction, but they actually did it, and they showed first time that it is possible to do the, using somatic cell, you can do the de-differentiation and you can do the cloning. So again, this just needs very basic techniques. It just does not need too much of your, high gadgets and technologies. It just need, understanding very well of how well you can take out these nucleus from the ova, how well you can culture them, how well you can control them, and then you can result into the full embryo development. A question there, yes. That's right. I think that's very interesting observation made. They could have used any of these two, right? They could have used any of these two to, for further growth. What in this case they wanted to ensure that their cloning has actually happened and it is actually not coming dictated by any of these two. So if you are using the third mother, now then you can totally claim that all the DNA content coming from the other two, it still is having potential to give rise to a new embryo. So that's what the good rationale of thinking about taking a surrogate mother. But of course this could have been done in the same sheep as well. So whether cloning is such a foolproof process, or is that like photocopying machine where you can make the photocopies of animals, you can keep producing whatever animal you like, having commercial values, then you can do anything for that matter, right? But cloned animals are not always perfect. They do not look exactly same, and these kind of observation people have made for many of the cloned animals, including cows, including cats. Let me show you one of the interesting example here. This cat is known as carbon copy or CC cat. It is also cloned cat. Now the two cats ideally should exactly look same because they are cloned copies, carbon copies. But one of the cat has some of the light yellowish tinge of the fur, whereas the other one has a grayish color. So whether this thing matters much or not, but ideally you do not have the same appearance. So while their genetic content could be still same and they have been derived from the same cloning process, but they're not exactly identical, right? So that leads some possibility that irrespective of how you are overcoming the, the natural laws of development process, but there might be some random events are happening, especially, X chromosome inactivation, etc., which could be leading towards, along with many environmental factors, which could be leading into different type of embryonic development for the different clones to happen. Let's look at, identical human twins example. So I'm sure you'd have encountered or seen some individuals who are twins, and they look very similar. They're very, very close in appearance, but they're not exactly same. And even their, kind of nature-wise and their property-wise, they cannot be exactly same. So it means it's not only the genetic makeup, it's also important that in which environment they have been grown and what kind of other factors, especially epigenetic factors, which are equally important along with the genetic factors, how they influence the development process. So therefore, two individuals, even two twins or two clones cannot be exactly identical. I think that has been one of the concern as well, that if you are planning to have at the commercial level, at the very large scale level, how perfect your technology is. Just imagine that you are doing mass production of certain leather or mass production of various products, right? Can you do the same way of mass production for cows? Can you do the same way of mass production for any of the animal which you want? Theoretically, yes, you can, but because you will not have success of producing everything perfect, people are not allowed to do those things at that level. So there has been many observations that many of the cloned embryos, they initially they look normal, but after some time they have encountered certain type of defects. Especially it was apparent in case of Dolly, which was the example which we talked. Even Dolly died in 6 years' time, which is not the right age for dying for a sheep. And people may feel that probably is the time that when the DNA was taken, and is the age being counted from that DNA, because that DNA from that sheep would have already taken 20 years probably, and then are you counting now 20 plus 6 years? So these are the kind of questions there. So therefore you are not starting from zero for again to reach that type of time life span. So Dolly early death was one of the setback for the animal cloning field, because then they cannot answer some of these kind of questions. And also many clones, they encounter a lot of abnormalities. So as I mentioned, are there certain epigenetic factors, many of the chromatin proteins which we talked little bit when we talked about histone proteins and how they are involved in the chromatin modeling part. So some of those, are they actually being involved for the reprogramming of the donor nuclei, and are they doing the chromatin restructuring, which is equally important along with the genetic factors. So some of these things have been in the field a limitation, a thought process against that, why you should not use this particular technology at the mass production level. It has good proof of concept that now we have understood the cell biology and the developmental biology so well that we can do these things, but it may not be required to attempt at that level. So that particular, how to identify the mechanism which are underlying for these deficiencies or these kind of aberrations are very crucial. Before we do that, we should not try to just attempt and keep making the clones, which will be all kind of having variety of issues. So as a result, people were actually, the cloning for the animals were banned in US and then subsequently many parts of the world. And then only thing which people felt the need for it, that can we use the cloning procedure for the therapeutic purpose? Can we take the cells, we change the cells for the requirement of what you need at that time? Let's say you need for your, somebody need for a heart failure that time, or for a brain disorder. Can you take these cells, embryonic stem cell, and then you can transform into the right type of cell which you want to convert, and use the cloning concept for doing that at the cell level, not at the full organism level. So understanding the cloning for the stem cell production was actually promoted and further continued for this field. Stem cell you are going to study in much more detail in the, once I finish my part, but in this context briefly, stem cells are those unspecialized cell which could be converted or modulated into a given specialized cell type if you provide them the right type of nutrients, right type of growth factors at the very early embryonic stage level. And it just shows you here that if you can culture these in different type of culture condition, the same embryonic stem cell could be converted into liver cell or nerve cell or blood cell. And I'm sure you all understand and agree to it that this concept has huge potential from your medical point of view, because one could use this for many deficiencies, many diseases. for replenishing the cells or the same individual, because if you are taking the cell or different organ from somebody else, then you will have always that the transplant tissue, the rejection, and then if you are growing the cell from the same individual and you are then transplanting back, they will have much more advantages of taking it back. So how to some scientists now then plan an experiment that how to deprogram, not reprogram, but deprogram the fully differentiated human cells into the stem cells. So while one concept is you take a stem cell, you are trying to convert those into a specialized different type of cells. Can you take the differentiated cells and now you are trying to make them as a stem cell? And this was one of the very classical experiment, not to the great extent I'm going to talk, but just to brief you, this is one of the Nobel Prize winning research done by Dr. Yamanaka, Shinya Yamanaka in Japan. When they took some of the precursor cells, made the skin fibroblast, and then actually they tried some transcription factors which are listed here, which are OCT3/4, SOX2, MYC and KLF4. By addition of these transcription factors, they were able to promote generation of induced pluripotent cells, and that was kind of one of the experiment which led them to win the Nobel Prize. So let me kind of take some time now and give you some perspective on how our understanding about the cell cycle, cell reprogramming, how it has resulted into big scientific prizes and some of the scientific failures and some of the scientific scandals. So I talked to, you may not recognize them. One of them is Sir John Gurdon, which is shown here. He did the experiment of frog cloning, which we talked in 1960s, 1962. And Dr. Shinya Yamanaka in 2005 or 6, he did this experiment of doing the cell reprogramming, especially stem cell experiments. And both of them, in some way, looking at different type of cell reprogramming, they contributed to the basic research of how the cell can be reprogrammed. And both of them made original contribution, one at the level of the animal cloning level and one at the level of the stem cell and understanding about the induced pluripotent stem cells. So both of them were kind of basic research, groundbreaking research, and both of them got Nobel Prize for their contribution in 2012.
Next are the 2012 Nobel Prizes. The first was awarded today for groundbreaking work in reprogramming cells in the body. Ray Suarez looks at those achievements.
The Nobel Assembly at Karolinska Institute has today decided to award the Nobel Prize in Physiology or Medicine 2012 jointly to John B. Gurdon and Shinya Yamanaka.
The two scientists are from two different generations and celebrated today's announcement half a world apart. But today they were celebrated together for their research that led to a groundbreaking understanding of how cells work. Sir John Gurdon of Cambridge University was awarded for his work in 1962. He was able to use specialized cells of frogs, like skin or intestinal cells, to generate new tadpoles and show DNA could drive the formation of all cells in the body. 40 years later, Dr. Yamanaka built on that and went further. He was able to turn mature cells back into their earliest form as primitive cells. Those cells are in many ways the equivalent of embryonic stem cells because they have the potential to develop into specialized cells for heart, liver, and other organs. Dr. Yamanaka is currently working at Kyoto University. Embryonic stem cells have had to be harvested from human embryos, a source of debate and considerable controversy. For Gurdon, the prize had special meaning. At a news conference in London, he recalled one school teacher's reaction to his desire to study science.
It was a completely ridiculous idea because there was no hope whatever of my doing science, and any time spent on it would be a total waste of time, both on my part and the part of the person having to teach him. So that completely terminated my science at school.
The men will receive their award in Stockholm in December.
Additionally, there are many scientists in the world who have been attempting to do the cloning experiments, and many of them were successful as well. One of them shown here is Dr. Wang Woo-suk from Korea, South Korea, and he made series of cloned animals. One shown is, along with him is a dog, but he made many cows which were all cloned, And in Korea, people felt that he's going to bring lot of wealth, lot of commercial value, and probably so much income to the country, because he has the capacity to do all this kind of cloning for their high yielding varieties, which can give them good yield for food, different type of products. He became kind of one of the big stars in the field, because he was doing series of these cloning experiments and showing one after the other they can clone many type of animals. And then they were also attempting towards the stem cell research, and showed some of their kind of experiment, especially one of them is known as somatic cell nuclear transfer, which was published in one of the big journal Science. It is first time that has the potential to do the human stem cell cloning as well. So after these research, and then kind of they publish these works. If you're not aware, in basic science field, the journal Science and Nature, they are the topmost journals, which are the highest impact factor journals. So you have a lot of difficulty in publishing these journals unless you have a sound hypothesis and good experiments and validation done for those experiments. So they did publish in Science. Dr. Hwang Woo-suk is the first author in this paper, along with many of these authors, when they are showing that somatic cell nuclear transfer is possible to be done. They also showed another paper that they could now do the cloning of the blastocyst, which has potential for the human type of cloning, human embryonic development, and again published in Science in 2004. So these two were kind of major breakthroughs in the field, and people were already finding him as authority in the field. So as a result, many people started trying to replicate their experiments, and then after some time, people wrote to journal Science that there are some discrepancy which we find in these experiments. We are not able to reproduce these results very well. I'm sure you cannot read it from your distance here, but some of the things which I can read for you. So Science Journal started investigation against this research and their work, and they mentioned, from the information that we have so far, it seems that they have made some honest mistake. We have no evidence that there was an intent to deceive, but they still kept the research on hold, and then they started investigating more. But many people have observed that many of the claims what they have done in the papers are actually not meeting to the right experimental requirements, and others are not able to reproduce those experiments. So after some time, their university started investigation, and then they found series of allegations and series of issues which are involved in these experiments. And as a result, you can see this news clip here.
Disgraced stem cell researcher Hwang Woo-suk has been sentenced to 1 year and 6 months in prison and 2 years of probation for embezzling state and private funds and buying human eggs. The Supreme Court issued the verdict Thursday, which comes 8 years after he admitted to faking his research. The ousted Seoul National University professor was indicted in 2006 on charges of fabricating the results of his human stem cell research and embezzling nearly $2 million in research funds from the government and two domestic companies, SK Group and Nongyap.
Well, so there was rise and fall of this chart. A lot of things happened from 2004 onwards. He was sentenced to jail. He again tried to make a comeback. Again tried attempting some experiment, but once people lose trust in you, lose confidence in you, it's very difficult to make the comeback. So he did try to show some successful experiment afterwards, but then there is no authority to actually accept those results. Another story developed with Dr. Haruko Obokata from Japan, and she was trying to she was a Japanese scientist like Dr. Yamanaka, and she was trying to see that can she work on the stem cell part and make the process of cell reprogramming much simpler. And she came up with a concept which is known as STAP, or stimulus triggered acquisition of pluripotency, where she said that rather than using those genes or transcription factor which Dr. Yamanaka showed for the Nobel Prize winning study, you can just simply change the pH of the medium, and just that pH trigger itself could actually lead to those gene changes which could acquire the pluripotency. And that was something which brought very novel kind of concept. And she published this work in another distinguished journal, which is Nature, which you can see here, and she is the first author of a stimulus-triggered fate conversion of the somatic cell into pluripotency. Another paper they also published again on the same concept in 2014. These are much more recent studies. Then people started following up, how simple this thing can be now, and can we reproduce those? And when they started reading their papers much more in detail, they found that there are a lot of issues in the images or in this particular paper. This lane looks like something has been taken from some other gel and brought in and put it here, which is not belonging to this experiment. They're looking at the DNA profile here. In this case, they're looking at a cell under microscope, and just by changing the contrast or brightness, probably you could make something look like this whitish tinge, which is not actually accurate. Or by inverting the embryo part of the images, you are making it look like this. So many of these observations were made. People started investigating against these, that many of these things what actually has been presented looks like has been played quite a bit of the image processing side, and they are not the accurate reflection of the what result one could obtain. So, with all of these issues, when their university started investigation again towards them, and that was much more serious investigation, much more recent thing, and let's listen what happened here.
Scientists from a leading Japanese research institute have apologized for the grave mistakes in recent papers on a new way of creating stem cells. The research was hailed as a revolutionary discovery when published in January in the British journal Nature, but mounting questions about the methodology and results forced the Riken Center to take action. NHK World's Takafumi Terui reports.
Riken scientists spent about 4 hours in front of the cameras talking about what has become an international scientific scandal. They say the findings of their investigation into the stem cell study in question were still inconclusive. They noted they've seen no evidence of deliberate data manipulation so far. But the president of Riken, who's a Nobel laureate in chemistry, acknowledged serious mistakes were made.
I would like to first and foremost express my deepest regrets that articles published in Nature by our scientists are bringing into question the credibility of the scientific community.
All right, well, so, when you are part of an ambitious project, sometimes you are competing for, something to make a device, something to make a product, something to compete for international competitions. You are part of a team, and that team is actually, trying to compete to be best in the world, right? In the process, sometimes you just get too greedy, and then you start thinking about some fine tuning, and that could lead towards some sort of fraudulence as well. And this is what has happened in these cases. And if you're part of those teams, you are equally, involved. Even if you are not the one who has committed the mistake, you will be still considered part of that team, and you have to own that particular problem yourself. So in this case, every author, even those who have not committed the mistake, even all those authors, they actually, were punished. They were all, removed from job. Their salaries were taken. Some of them were sent to jail. This is a Nature Journal wrote what the punishments were there for all the scientists involved in the STAP research. So every scientist was actually, who was involved in the paper, even if they may not be aware that something like this fraud would have happened, it's your responsibility as a part of that team to ensure that you're also confident about that work and that research which is happening. So it's very crucial to just get the, when you are trying to get the glory, you have to also own the failures of that team. So, but what was the most, disheartening to see, one of the researchers committed suicide after this particular failure and this particular retractions. In the same lab, they committed suicide, and this was very, sad to see that the fate of the full team and what has happened to that. So, if I just kind of summarize this part, although we are talking about cell reprogramming, but it's always something to learn, and those lessons could be much bigger than just learning development. So, cell reprogramming, we talked about some of the basic fundamental research which gave into the Nobel Prizes. We also seen that, some of the fall of the star, which tried to reach to the same level but use some sort of fraudulent ways of reaching there, and that was not so appropriate. So it just brings, I think last 2 minutes or so I'll take to summarize that you need to follow some ethics in research and publication, especially in the kind of profession we all work, whether you are engineers or somebody is, biologist or medical practitioner. Society has a lot of expectations from us, and they do believe that whatever we report is actually very accurate. So I think your devices, your models, your analysis has to have those values, those meanings, and then only I think, people are going to have trust in us. And as I mentioned that many times, you are all going to compete for many of the competition at the world level. Please ensure that you are following the ethics in whatever work you do, and that's very crucial not only to study development but also for your day-to-day kind of life achievements. So today's lecture, we try to understand some of the phenomena involved in this cell reprogramming. Many elegant experiments done for the cloning, especially different type of animals were cloned, and later on stem cell cloning was also performed. We then tried to take a case study and interesting story to see that how different experiments or the major breakthroughs which were reported in the studies, some were based on the sound understanding resulted into the Nobel Prize. Some of them were also fraudulent research and were actually penalized and became one of the scientific scandals of the field. I think it's important to understand that this concept of cloning is, on one hand it's like a scientific fiction for us, which looks like getting true. At the same time have many ethical issues involved, because when to clone, why to clone, and what impact it may have for the society. At the same time, from the case study you would have witnessed that what practices one should follow in doing the scientific experiment is very crucial. So let's continue some of these discussions about the science ethics involved in doing this kind of experiment and what to be reported in research and publications. Let's continue that in the next lecture. Thank you.